And it's my pleasure. I'm Sarah Linklater. I'm Chief Scientific Officer with JDRF Canada, and delighted to be co-hosting this event today with Health Canada. Please do keep yourself on mute. I need to let you know we are recording this event. If you would like to access simultaneous interpretation, please access this at the bottom of your Zoom screen by hitting simultaneous interpretation or the 3 dots that will allow you to choose your language. We have 4 presentations. We're going to hold questions and discussion till the end. I do hope we still have some time. Verbal questions will be prioritized if you use the raise your hand feature. You're also welcome to use the chat. So without further ado, I apologize for the brevity here. I'm introducing Bruce Perkins, Professor of Medicine and Clinician Scientist from the University of Toronto, who will speak on adjunctive to insulin therapies. Bruce, I'd welcome you to share your screen and present to our group today. Thank you so much.
All right, I will give a briefer, but in maybe even more sophisticated introduction that I tried to give a few moments ago. Sarah, thank you so much. And members of Health Canada here, we have huge respect for you. We wish we were meeting in person. I've really enjoyed any interactions I've had at sessions like these in the past. So I'm going to be speaking around adjunctive to insulin therapies in type 1 diabetes. And I'll just disclose that though I try and limit my interactions with industry, I've served as a speaker, and I've had some research support and served as an advisor, particularly with Boehringer Ingelheim, whom I've worked to develop a phase 3 program in this domain of adjunctive to insulin therapy. I'm going to cover it in 5 steps. They're here. Sarah, do we have the translation now or no? Or do you know?
We do have the translation now.
Okay, well,
so that's running. Please feel free to proceed.
To the translators, I apologize. I've been
speaking. Au traducteur, je suis désolée, je parle tellement en vite.
You've had a moment to look at that, but I'm going to begin with context and strategies. It's 100 years ago that insulin was discovered, and Banting and Best today would be impressed with many innovations, but they'd really be hit by the fact that many people with type 1 feel isolated. They carry huge amounts of self-management burden, and of the evidence-based targets that we set for blood sugar control, it's only, and we know this in Canada as well, only 1 in 5 people who are meeting those targets, even with the sort of therapies that we have available today. This is a major problem. So to overcome this epidemic of high blood sugar, we have a number of innovations that we work with. Multiple daily injections, if we use insulin pump therapy instead, or supplement it with continuous glucose sensor data, or that if we harmonize these 2 things into automated insulin delivery, the so-called artificial pancreas system, evidence shows this, that on average, we can improve an A1C by about 0.3%, which is a very important mean improvement, but obviously wouldn't take away the very high A1Cs that we're seeing in this population. The idea and my philosophy is that the more choices that we have available to people, those who, for example, start pump therapy and have more than a 0.3% A1C improvement, know that they found a therapy that can help them reach targets, whereas someone else might focus on glucose sensing and maybe undertake pump therapy as a choice later. These are examples. And I'll just tell you to zip ahead that of adjunctive to insulin therapy, so medications used to help insulin work better, to struggle with bringing blood sugar down to better levels. 2 have been approved in other countries, not in Canada, pramlintide in the US and SGLT inhibition through Europe and Japan. And these actually have trials that show A1C improvements that are in the same ballpark, if not even numerically better than what we're seeing with these amazing therapies that we've adopted, like pump therapy and sensors. So let me just give a little guide to what's been studied. Amylin analogs, or this is the pramlintide I mentioned. Amylin, it's mimicking amylin produced by beta cells, has an incretin hormone-like effect, if you will, delays absorption of carbohydrate and improves postprandial blood sugars nicely and can improve A1C nicely. Approved in the US, uptake has been low, only in about 1% of people. It's limited by hypoglycemia and the burden of 3 extra injections a day. But there are novel ways to help overcome these issues that you'll be hearing from Ahmed Haider about, integrating it in a automated insulin delivery system. So I'll leave that. Now, all these medications have been transformational in type 2 diabetes, improving metabolic control. And if they work so well and are so transformational in type 2, why on earth would we not aggressively test them on top of insulin in people with type 1 diabetes, given that we're facing such an epidemic of high blood sugar, high A1Cs. So for example, metformin and DPP-4 inhibitors, they've been extensively studied. They don't actually have a mean A1C improvement. The GLP-1 receptor agonists, these are the injected forms, well, the ones that have been studied are injected forms in type 1 diabetes. They do have a bit of an A1C improvement, but the companies that developed them didn't move forward for type 1 diabetes indication, thinking that the A1C improvement may not be enough and there's some other complexities to the therapy. And there are other things, we use glucagon and its analogs in automated insulin systems and research, and things in development in animal models include somatostatin receptor antagonists and leptin that may have some benefits, particularly with prevention of hypoglycemia. But the class of drugs that have had the most traction in these last years is this class called the sodium glucose linked transporter, or SGLT inhibitors. And so I'm going to focus on this area. What these medications do, if you can picture the kidney, all of our kidneys, whether we have diabetes or not, put lots of glucose in the urine, and we have a mechanism to pull that glucose back in. If we didn't have that mechanism, we wouldn't have survived evolution, losing calories so early. People with diabetes, or even some high sugars, so people like me, I live with diabetes, we upregulate these SGLT inhibitors to capture the higher amount of glucose lost in the urine. The issue is that this mechanism is also pulling a sodium molecule with each glucose molecule. So us with diabetes, we tend to have a little bit of sodium or fluid overload as well. And so by blocking this exaggerated pathway, these upregulated receptors, in people with diabetes, it's a mechanism to help lower glucose through an insulin-independent mechanism, lose some calories that can have effect on counteracting the issue of weight gain in type 1 diabetes, which is a major gap. And finally, normalizing the sodium loss, which can help with blood pressure and a number of other factors. So that's the concept of SGLT inhibition. Well, in type 2, I said they've been transformational for blood sugar control, but because they needed to undergo large-scale safety studies to make sure that drugs in type 2 are safe for cardiovascular disease, this is the first class that was really shown to have huge effects on cardiovascular disease, and in this particular study that I'm showing here, big reductions in even all-cause mortality, and no one could argue that there's no more important clinical outcome than improving all-cause mortality from a medication. But above and beyond this, these medications now in other trials have been shown to reduce substantially chronic kidney disease, major outcomes, heart failure outcomes, and in fact, even heart failure reduction in studies in people without diabetes. And these studies are going on for chronic kidney disease as well. So really a major class of drugs that we have to treat seriously and consider potential long-term benefits in type 1 diabetes patients. If you don't mind, just give me a minute to kind of explain where my whole interest in this area started. So actually, it's close to 10 years ago, grouped with a great collaborator, David Cherney, to look at the effect of these drugs on kidneys. What on earth are they doing that could potentially protect or even harm kidneys? We didn't really know then. And we decided we're going to study it in people with type 1 diabetes who had never received this medication before. And we studied them over a few months. Here I'm showing an image from a study published just a couple of years ago where they did a similar experiment, but in animals where they could visualize. So the bottom line is that in people with diabetes, losing this extra glucose in the urine and upregulating SGLTs, the transporters, they pull sodium back in and it tricks the kidney into thinking there's very low sodium in the system because now the sodium's been taken out of the urine. When that happens, the kidney thinks that they're in a situation of dehydration. And what it does is it increases the caliber of the inflow blood vessel called the afferent arteriole to increase blood flow into the kidney filtering unit that you see here with these arrows. But the blood pressure is not low. It's just that the glucose is high and tricking the kidney into thinking the blood pressure is low. And so it increases blood flow into the glomerulus, the filtering unit, increases the pressure and causes long-term damage. We found that when we give this medication, we normalize the caliber of that afferent arteriole and decreases the intraglomerular pressure. So this is actually all driven by the normalization of sodium in the urine. And in fact, that sodium component has a number of mechanisms around blood pressure and cardiovascular and renal outcomes. So when we finished that study, we found, like, just quite frankly, even though we saw beautiful metabolic effects over the few months in our type 1 patients, many of them just did not want to come off the medication. We really pushed ultimately for large-scale clinical trials, which were performed. I could show you many of these. For example, there's almost 3,000 in the sotagliflozin program, over 2,000 in the dapagliflozin program. It's been studied in ipragliflozin, and here is the data for empagliflozin, and they're all quite similar. Here we had a run-in period to optimize insulin. We did a better job with insulin therapy to bring people's A1C down by 0.5%, but being randomized to this drug, empagliflozin, improved A1C a further 0.5% above and beyond placebo and seemed to generally be maintained over a year. Improved time and range. There was dose reduction in insulin, weight reduction that was substantial, even though these were only only half of people had overweight or obesity. Blood pressure reduction without an increase in severe hypoglycemia, despite the improvement in A1C. And in fact, minor hypoglycemia was reduced as well by 30%, particularly during the night. So really, unquestionably an amazing biological effect from these medications in type 1 diabetes. And if we look at all the trials, we get sort of a meta-analyzed A1C improvement of about 0.4%. So the elephant in the room, though, is that these trials were shown to have side effects. And a common side effect we see are genital tract infections that we see in type 2. Minor, we're able to manage these. But in type 1, what showed up in those trials was an increased risk of diabetic ketoacidosis. We know that DKA occurs at a rate of 5% generally in the population. That's 5 events for every 100 people we follow for a year of DKA. And these drugs seem to increase it. It doesn't seem to, we would call this a component cause, meaning when the events of DKA occurred in these trials, there was always an explanation, a pump failure, an intercurrent illness, but it seemed that these drugs accentuate tipping people into DKA. So we call it a component cause. The drug is neither necessary nor sufficient for DKA, but helps to promote episodes of DKA. We also found that it modified the presentation. Usually in DKA, we see very high blood sugars, and here we don't see as high blood sugars. So it changes the way it presents itself. The magnitude of risk was about a 2.5 to 3-fold increase in DKA risk. And if we look at the absolute risks, it would mean a number needed to harm of about 45 to 50 people. In other words, we need to treat 45 or 50 people for 6 months to a year to have one excess case of DKA, and that is too much. So we need to be able to overcome that. So how would we overcome that? Well, one way is just not use it. For example, as a regulator like Health Canada, don't approve these if submissions come through, and then there wouldn't be this accentuated DKA risk. So that was the tack taken by the FDA in the US. There were 3 applications and they rejected all of them for concerns over not being able to define the bounds or the certainty around that DKA risk, and also not having formally tested a mitigation strategy. Whereas the European Union, the countries in Europe did approve these, and so did Japan. So there are different tacks we could take. The second is we could select a lower risk patient subgroup. And in fact, where these have been approved, it's been approved in those with a higher BMI. People with higher BMI, 27, which is actually in the overweight range into the obese range, tend to be on higher insulin doses. And I won't show you the data, but the drugs that have been approved, they showed would appear to be numerically no increased risk amongst that subgroup. And so, for example, in the UK, the NICE guidance, they say that people should be under specialist care, have good education around DKA, and should have a BMI above 27. The other thing is we could implement mitigation strategies, and actually there's already been publications on simple rules of thumb, the stop DKA protocol or the stitch protocol. The idea, if you have a sick day feeling unwell, regardless of sugar, check a ketone. If elevated, hold a medication that day, extra fluids, food, and insulin, and restart when the illness recovers. And actually, you know, these have not been formally tested, although I will say that in the US, where there is a fair amount of off-label use, when these drugs first became available for type 2 diabetes amongst those with type 1, there was a much bigger risk of DKA. But once clinicians and people with diabetes started understanding this issue, you can see that even without, you know, publishing these mitigation strategies, the risk of DKA was not different than the general population with type 1 diabetes over time. The final mitigation, which is the one our program with empagliflozin we pushed for with the FDA, when using a dose that's a 1/10 of the amount of the typical dose in type 2 diabetes, so a 2.5 milligram dose of empagliflozin, we saw we saw improvements in metabolic profiles, not as much as the high doses, but still very clinically relevant nevertheless. And the reason we went for this dose was because in phase 2 trials, we saw that the amount of glucose excretion, and a measure of the effect of the drug, with that tiny dose was as much as you would get with the full dose in people with type 2 diabetes. And so there were sort of 500 people in this arm of the study, and rather than having the threefold-ish increase in DKA, we didn't see an increase in DKA in those receiving the smallest dose. So we saw this as a further mechanism for mitigation, having a type 1 diabetes specific dose. Finally, I'd just like to say that we've, in partnership with Ahmed Haider, who you'll be hearing from in a moment, we thought, you know, artificial pancreas, under the philosophy of multiple choices that people could have, we want to know the interaction of an artificial pancreas technology and these medications. And the issue with artificial pancreas now is that even though we can really achieve good, stable overnight blood sugars, once people eat meals, it's very difficult for these algorithms to handle them currently. And we thought that adding a medication that might attenuate the rise with a meal, like an SGLT inhibitor, that it might help the algorithm work better. And in fact, this study in Press was a daytime study that showed a 20% improvement in the time in range. And we now are completing this year a 5-month outpatient study as well. So let me just sort of give you my key messages. I think, Sarah, I've kept to time. You know, there is an epidemic of high blood sugar exposure in type 1 diabetes. Adjunctive to insulin therapies have a strong biological rationale. They've been transformative in type 2 diabetes, but they make sense on top of insulin in type 1 diabetes as well. They have this potential for even non-glycemic mechanisms to prevent long-term complications that are extremely important to people with type 1 diabetes. But for our evidence base, they have been extensively studied and they have clear metabolic benefits. Diabetic ketoacidosis risk exists, and I didn't speak much around this, but we need to understand that we've brought in therapies that increase DKA risk in the past, and actually insulin pump therapy would be a key example. But over time, we learned how to manage this clinically, had better cannulas and had better clinical management that we could overcome it. It would be a shame to lose this opportunity for the metabolic benefits of a class of drug, knowing that we could mitigate the DKA risk associated with it. And finally, international regulatory bodies have made different conclusions about risk and benefit. For example, the FDA has uniformly rejected applications, whereas Europe and Japan have not. And our hope is that Health Canada would consider this kind of therapy to really help as another measure to advocate for the health of people with type 1 diabetes in Canada. So I will stop there. And thank you very much.
Thank you, Bruce. That was really interesting, and apologies for the time pressure you may have felt there with us starting late. So we are going to hold questions until the panel discussion at the end, which might be shorter than planned, but feel free to enter questions and comments into the chat as we roll on if you would like. So now to introduce our second speaker, Dr. Ahmed Haidar, who's assistant professor at McGill University in Montreal, and he's going to speak to us about advances in closed loop therapy for type 1 diabetes. Thanks and welcome, Ahmed.
Thanks, Sarah.
We can see your slides now.
You can? Okay, thank you. Hello, everyone. It's my pleasure to be able to present today, and thank you for the invitation. So these are my disclosures. In particular, I work with Eli Lilly to commercialize an automated insulin delivery system. Yeah. So what is closed loop insulin therapy? So it's a novel therapy that is composed of 3 components. So the first one is the continuous glucose monitoring system. The second one is an insulin pump, and the third one is a mathematical algorithm that adjusts insulin delivery automatically based on sensor readings every 10 to 15 minutes. So the idea is simple. It's been there for a long time. So the first attempt was in 1960, but we really didn't have the ability to commercialize these systems or to bring them to market because we did not have the ability to measure sugar level in a continuous, non-invasive manner up until 10 or 15 years ago. So even though the idea was there and you see big literature in the 70s and 80s, our ability to actually build portable devices that are usable in clinic became only possible in the last 10 or 15 years. So there has been multiple devices in the markets. There is 2 in Canada, 2 in the US, and 2 in Europe, and there are others that are under development. So this is just quick summary of multiple randomized controlled trials from Tandem, from CAMP APS, which is a system developed in Cambridge, from Medtronic, Open APS, and so on. Now you can see that the improvement these randomized controlled studies showed in the time in range, which is the percentage of time sugar levels are between 3.9 and 10, is around 11%. And this number has been consistent coming from multiple studies. So it looks like we're reaching some form of saturation in terms of the improvements that we can get from these systems. And what I'm going to talk about today is that what is next. So we saw multiple systems coming. All of them are showing around 11% improvement, which is 2.5 hours per day. And what will be the next step? And what is next? I don't really mean next year. It's more the next maybe 10 to 15 years. So the first thing that on average, it depends the type of people you recruit in your studies. But on average, what we see that this system achieve around 70 to 71% of the time in range. Now, the current recommendation is that people with type 1 diabetes should try to have their time in range above 70%. But when your average is 70, that means half the people have a time in range below 70 and the other half have above 70. So we still have significant work to be done in terms of improvement in time in range. Now, the other thing, all the systems are hybrid. So hybrid means with every single meal, the user have to do carbohydrate counting and they need to enter that number into the pump, or if the algorithm is running on a phone, they have to enter that number on the phone, and then they get insulin boluses. So these systems, really what they do, they modulate what we call basal insulin delivery. They don't touch the bolus delivery, but the bolus is around 50% of the total insulin delivery. So what people are looking at is how we can alleviate the need of carbohydrate counting, because this is a burden to the patients, and there is some literature that shows that it can induce, for example, unhealthy dietary choices and so on. Now, the third component where people are looking at is that you're wearing really 2 devices. So people have to wear a glucose sensor, an insulin pump. If the algorithm is running on a phone, they need to have a phone with them. So it's a little bit bulky device. So people are looking at how we can make these devices smaller, how we can integrate devices together. I mean, you call people, you tell them, I mean, in our studies, we're running a study with an artificial pancreas system, and they come and you give them 2 pumps and a phone and a sensor, and they were like, wow, this is not what I thought about when you told me artificial pancreas. So I think we need to make these systems smaller. So how people do this, or how people are trying to do this. So one of them is that we try to develop faster algorithms or better algorithms. And I would categorize these into 2 different categories. So one of them is fine-tuning the current existing systems. And this is recent data that is just came in the Lancet from Medtronic, where they compared the 670G, which is their commercial automated insulin delivery system, with an advanced system that I think will come to the market soon. And they showed that they were able to improve time in range by around 4%, but you can see they had improvement across the entire time range of the day. Now, the other one where people are talking about is what we call cloud-based day-to-day learning, because now all these systems, when they make decision about insulin delivery, they only look at the previous few hours. So is there any meal in the last 4 hours? Is there any boluses for the last 4 hours? And then they make decisions for the future. But you can think about different type of algorithms. They look at a week worth of data. And then these algorithms will make adjustment to, for example, the aggressiveness of the system or the insulin to carbohydrate ratio. So we have done some work, us here in the diabetes camp, where we put people on a system for 8 days, and we were able to have an algorithm that automatically adjusts the beta rate and the carb ratios. And you can see for some patients, we have to make changes to the carbohydrate ratio in camp setting by as much as 50%. Now, this data came from Tandem recently, where they didn't test an automated way of adaptation. They tested a manual way of adaptation to see how much improvement you get when you make adjustment to some of the algorithm parameters. Now, our data and their data showed that potentially we don't get really much improvement in overall time in range. The benefit could come more like a local benefit. So if someone is having a hypo after breakfast, if you make adjustment to the carb ratio at breakfast, you could improve at breakfast, but unlikely you get improvement for the entire time in range. So some regulatory questions. So if you have a small increment to the algorithm, do you really require a pivotal study? I actually don't know. Because when I work on algorithms, sometimes I look at changes. I know this is minor, but do I really need to go and run a 300 patient study, or I could just run a 20 patient study? And I think it's, as a field, we still don't know the answer for that. Now, can we use computer simulations to accelerate the approval process of incremental changes to the algorithm? So maybe you could do extensive simulations, and then you can do a 20 patient trial instead of 100 or 200 patient trial. Now, I personally am a bit skeptical about simulations. I think they're useful, but I mean, I think I don't think they replace clinical testing, but some people do believe, and FDA actually seem to believe in simulations. Yeah. So the other approach, which is more to build on what Bruce mentioned, is to add drugs to closed loop systems. And there has been multiple studies. So the faster insulin, so people have done studies with the FIASP, with Lunjov from Eli Lilly, and so on. You could look at the glucagon. So there's a company trying to develop a dual chamber pump where you don't only deliver insulin, but you also deliver a glucagon. And there are a few companies developing stable glucagon compatible for pump use. And this is data from one of our studies where we compared sensor-augmented pump therapy, insulin alone system, and insulin glucagon system. And here we have an outlier, but you can see that in most people, you can reduce the risk of hypoglycemia further by giving glucagon. You could look at pramlintide, and the way to do it, we believe, is to have a co-formulation where you combine insulin and pramlintide in one drug. You put that in the pump, and you combine them with algorithm, and you can get potentially more benefits. And there are a few companies looking at a co-formulation of insulin and pramlintide. There are SGLT2 inhibitors, as Bruce showed earlier, and there's GLP-1. So GLP-1 studies has been mostly with short-acting GLP-1. There hasn't been any studies with long-acting GLP-1. We're looking our group to do a study with semaglutide in closed-loop systems. So again, regulatory questions. Does the new labeling to include the newer insulin require a pivotal study? Because with pumps, if you have a pump approved for novo insulin and then you want to add FIASP, you probably have to do just compatibility testing. But if you bring a new insulin and it interacts with the algorithm, and the algorithm design may not be appropriate for that insulin, so how do you handle this? Do you have to go and run again a 3,000 or 300 patient study, or you can keep things faster? Again, simulations, can they be used in this? And the other thing is that what is the process to approve adjunctive therapies on top of closed loop therapy? Because some of these algorithms may not be appropriate for some of the drugs. If you talk about SGLT2 inhibitors, then you have a system that suspend insulin for 5 hours. would that be appropriate if someone is on SGLT2 inhibitor? So you really need to think about not only the drug and the algorithm alone, but how they interact with each other. Now, the other thing I mentioned is alleviating carbohydrate counting. So there are multiple incremental approaches to try to alleviate this burden. So one of them is to ask the patients to do what we call simple meal categorization. So to choose if the meal is small, medium, or large. The second, maybe less simple system is just simply to do a simple announcement. And maybe the ultimate system is that people don't do anything. They just wear an insulin pump and a sensor, and they eat whatever they want, and they don't have to worry about any insulin boluses. And current systems are all hybrid. Some people are looking at meal categorization. We see more studies in the literature about meal announcement, and we have seen only a couple of studies with full automated system, but the field is moving in that direction. And this is a recent study from a group in the Netherlands where they tested insulin and a glucagon system, which is fully automated. And you can see that the time in range was better with sensor augmented pump therapy. And they had no carb counting, no simple meal announcement. It was completely fully automated. So again, a regulatory question is how much degradation in glucose control are we willing to tolerate to alleviate carbohydrate counting? And the reason this question is relevant because alleviating carb counting is easy. You tell people don't do carb counting and that's it, but then you end up having A1Cs in the 10s and maybe higher. But alleviating carb counting without degrading glucose control is difficult. But what we are seeing now in the data and the literature is that maybe you degrade a little bit of control and you're able to alleviate carb counting. And this is data, unpublished data from our group where we do FIASP and a pramlintide full automated system compared to the insulin alone system with boluses. And you can see that when we have a fully automated system, we lose a little bit control in the early postprandial period, so 2 hours after each meal. But after 2 hours, because we give pramlintide and FIASP, we bring sugar level back to target. So the question we have is that if you submit this to FDA or Health Canada, we tell them, we have a therapy. People don't do anything around meals, but you lose a bit of control in the first 2 hours. How do you feel about it? And I don't think we have answers at this point. So I want to talk briefly about study we're running, which is funded by JDRF. It's 2 weeks per intervention, 15 adults, 15 pediatrics. In one arm, we have FIASP and pramlintide with simple meal announcement. So the people will have 2 pumps because we don't have co-formulation. So we're mimicking the co-formulation by delivering the 2 drugs in 2 pumps. But people simply press a button at mealtime and that's it. Now we have a FIASP and placebo system with simple meal announcement. And the third arm is FIASP and placebo with full carbohydrate counting. Now we have 4 pilot participants. I decided to show quality of life data. So only this is only the first 4 people. So we had the diabetes distress scale, and the baseline was 2.3, which was moderate distress, with the carb counting system 2.4, with the simple meal announcement and pramlintide, we brought it down to 1.8, which is little to no distress. And with simple meal announcement and placebo actually went up because when you don't have pramlintide, you alleviate carb counting, but you lose the glucose control. When you look at emotional burden subscale, it was 3.2 at baseline, so it's high distress. Again, only 4 participants. The main study is still ongoing. 3.4 with carb counting. With simple announcement, FIASP and pramlintide, you brought it down to 2.4. So we moved from the high distress to the moderate distress. And simple announcement, we went up again to 3. So we're hoping to finish the study by maybe August. So hopefully we will have strong data at that time. Now, I just want to put some quotes from the participants who did the study, because we did with them semi-structured interviews. So this is one patient. She said, I just found that we are all in a complicated situation right now. She's referring to COVID. Not having to worry about what I was eating took such a burden of my diabetes, took such of a burden of this huge thing in my life. I just felt more relaxed about that part of dealing with the disease, which is a pretty significant part. Another patient said, you can eat whatever you want, whenever you want, and that's neat. I'm like a picker, and it's always been hard to control diabetes as a picker. It really is the best, the best. Now, the third patient was a bit interesting. He said, I feel a little bit out of control at first because suddenly we're telling them, don't worry about your meal, just press a button. I had to get used to that, not having to do a self-calculated extended bolus. I kept the worry about it, but I found it generally did a really good job of taking care of me. And I think this shows that by alleviating carb counting, we're actually inducing some cognitive hopefully positive changes on the patient, that they feel maybe a little bit out of control, but eventually they will feel less burden. So we're looking at quality of life with surveys and questionnaires and interviews, and we plan to look at this data carefully. Now, this is my last slide. I don't do hardware, so I don't have much to say, but again, we're looking at smaller devices. I took this picture from the internet from, I think someone posted about the Dexcom new sensors. But you can see these devices will become smaller and smaller. I think we need to integrate pump and sensor together at some point, potentially in fusion set at the same place where you actually measure sugar level. There are some startups are looking at this, and these, I think they will be coming gradually to the market. And thank you. This is our team, and apologies if I was a bit fast. I'm a bit worried about time. So hopefully I was not too fast.
Thank you, Ahmed. That was really wonderful. And you have made up some time, you and Bruce together. So thank you for that. We're running about 10 minutes behind, but we should still be able to get everything that we'd like to do done. I also really appreciate the patient quotes. Those are always a really wonderful complement to the clinical data that you shared. So we're going to move on to our third speaker now, who is tackling a different topic. Our next speaker will be Dr. Gregory Corbett, who is Professor of Surgery, Scientific Director of the Alberta Cell Therapy Manufacturing Facility, and also Director of the Alberta Diabetes Institute of Histology at the University of Alberta in Edmonton. Dr. Corbett will be speaking on advances in beta cell replacement and how the field is closing in on a functional cure for type 1 diabetes. Welcome, Greg. We can see your desktop and slides are coming. That looks good. Thank you. Greg, we can't hear you. We don't have audio. Nope. We see you, we see your slides, but we can't hear you. Still no. Despite all our tech checks, sometimes this happens. Greg, I wonder if you want to log back out. Oh, something's coming. Try again.
Hey, can you hear me now?
Yes, yes, that
Okay.
works.
So I'm going to share my screen again.
Okay.
Okay.
That looks good. Here we go.
Okay. So sorry about that. I'm not sure, but that's typical. Okay. Can you still hear me?
Yes, we can.
Good. Okay. So I'm going to discuss about the current advances in beta cell replacement. So my disclosures, I consult for Sema Therapeutics as well as eGenesis. As we've been discussing, we know that the treatment options for patients with type 1 diabetes is the complications of suffering from hypoglycemia and the potential side effects of hyperglycemia. So we need to find better treatments for having better glucose control in patients with type 1 diabetes. So as we just heard, there's quite a bit of advancements in the artificial pancreas and the closed loop systems with multiple Canadian leaders. And another method of treating patients with type 1 diabetes is beta cell transplantation. Many of you may have heard about the Edmonton protocol, where we published in 2000 getting 7 patients off daily insulin with good glucose control following clinical islet transplantation. However, there are a number of considerations and obstacles that we encounter with islet cell transplantation. One of the main things is the effects of long-term immunosuppression. Because these patients are getting tissue from an organ donor, they have to be immunosuppressed to prevent rejection. And the amount of immunosuppression we use in clinical islet transplant recipients is quite high. It's actually much more aggressive than in a liver transplant recipient. So we're very limited to the number of patients that we can transplant because we gotta weigh the risks of the immunosuppression versus the benefit of the transplant. We also know that immediately when we infuse the human islets into the patient's liver, there is major inflammation. We lose approximately half of the islets within the first 24 hours because there's a significant inflammatory response. So if we were able to overcome the risks of immunosuppression, and we may be able to transplant more patients, but then we would have to find an unlimited or a greater supply of insulin-producing beta cells because we're limited by the number of human organ donors. So that is the main topic of my talk today, is the beta cell source, because there is a shortage of human organ donors. But we also know that sometimes the quality of islets that we get from a human pancreas is not very good, and sometimes it doesn't function that well after transplantation. So what are other potential sources? Well, the field of stem cells. We can get stem cells from embryonic stem cells. We can generate them, you can culture them, and theoretically, they can be differentiated into many different tissues, including insulin-producing cells, and these can be used to transplant into patients. However, there are ethical issues with embryonic stem cells, so the field has really been pushing for pluripotent stem cells. So it's the same thing. We have unlimited supply of these pluripotent stem cells, and theoretically, they can be differentiated into different types of tissues. So how do we make pancreatic cells? We have a stem cell either derived from an embryo or a pluripotent stem cell, and we try to differentiate them in culture, in vitro, so that they have a lineage into pancreatic cells, specifically insulin-producing cells, and we kind of stop it so they don't differentiate into other cells. So this is the whole idea: is you take a stem cell, can you make insulin-producing stem cells from that? So stem cells to beta cells. It's a multi-step cell manufacturing process. So you start with your stem cells, and you have to expand them to make what's called a master cell bank. This is an expensive procedure. You require highly qualified personnel, many different reagents. So this is an extremely expensive process. And then you differentiate them into the insulin-producing cells. You also need the appropriate infrastructure to meet FDA and Health Canada's guidelines. This has to be done in a facility that's doing good manufacturing practice. So where are we right now with this? Well, the production of clinical grade stem cells that are being done, the current companies is ViaCyte. They have done a phase 1 safety and efficacy trials here in Canada, in Edmonton with James Shapiro, and in Vancouver with Tim Kiefer. There's also the company Vertex Pharmaceuticals. They bought out Sema Therapeutics for about $900 million. So this is another major company. And Novo Nordisk is another company that is investing a lot of funds into deriving insulin-producing cells from stem cells. So ViaCyte is the one company that is undergoing phase 1 clinical trials. They're located in the San Diego area. So they have developed a number of protocols. They have a lot of patents behind it on procedures or techniques that you can differentiate embryonic stem cells into insulin-producing cells, and they're undergoing clinical trials. So this is an example right here of a video taken from Edmonton. These embryonic stem cell derived beta cells are in a device, and this is placed, as you can see, subcutaneously in these patients in this phase 1 trial. Health Canada and FDA, their regulatory requirements was that it has to be in a retrievable device, because if you have undifferentiated cells in these preparations that are going into these patients, that could form tumors. So this is the concept behind it. So you can see here in the video that they're being placed underneath the skin. And the purpose of this phase 1 trial is, can the cells survive and is it safe? So you can see this is some media publications from ViaCyte, and they're showing that when they retrieved these stem cell derived cells that were placed in these pouches underneath the skin of these type 1 diabetic and controlled patients, they reported that the cells survived, and they had a lot of insulin, glucagon, and somatostatin positive cells surviving. However, I think this is a snapshot of one of their better results, because I think that what they found was their first phase 1 trial, the cells were placed in this encapture device. So it was completely closed in here. And the concept was that blood supply would form on the outside of this device and allow the cells to survive. However, I think what actually happened in a lot of these patients in this phase 1 trial is there was a lot of foreign body response and fibrosis, and a lot of the cells did not survive. So the next phase approach was to take these devices and put holes in it so the blood vessels can grow into it. Because it's not a completely closed system and there's still a possibility that tumors can form and escape systemically into patients, these patients were also given immunosuppression as a safety measure. So this is I think is the only clinical trials that are being done with beta cells derived from embryonic stem cells. So the other thing is how do you manufacture stem cells into beta cells? It's a multi-step manufacturing process, and it requires a lot of expense to do it, and you need an appropriate GMP infrastructure to do that. So here in Edmonton at the University of Alberta, we have what's called the Alberta Cell Therapy Manufacturing Facility. This is a GMP facility that is able to produce clinical grade cells. We got money in 2009. We got 10.6 million from CFI. At that time, the Alberta province matched that 10.6 million, and the U of A put in approximately 5 million. It's a 10,000 square foot facility. It's the only one in Western Canada. We have currently used bone marrow-derived mesenchymal stem cells to go into a phase 2 trial to treat patients suffering from sepsis. We are making CAR T cells. We're going to actually next month do our first patients. We have created a hepatitis C vaccine for Michael Houghton, who just won the Nobel Prize in medicine. And we just recently completed a COVID vaccine that's in Ottawa right now for fill finish. And because we have the highly qualified people who have the regulatory expertise, we're providing support to other Canadian islet beta cell researchers. So this is an example. This is the floor plan of our facility. It's 10,000 square feet. Highlighted in yellow here, you can see we have 6 clean rooms to produce different cells. And it's actually quite a state-of-the-art facility where we're able to produce insulin-producing cells from stem cells. And this is a project that we're just getting underway with James Shapiro. So we have the infrastructure here in Canada to make insulin-producing cells from stem cells. So basically, what is the pathway to the clinic? We do our basic research, and we have to do preclinical studies to apply for a clinical trial application. So there's a lot of debate in the field is what are the preclinical studies? First studies are usually done in rodents and mice. ViaCyte did their first preclinical safety studies in mice, and then when they went to move to rats, they had a lot of problems because there was significant foreign body response. So what are the regulatory requirements to bring stem cells into patients? Do we need to do non-human primate studies for safety preclinical studies, or is it good enough just to do studies in rodents? So besides getting insulin-producing cells from stem cells, is there other alternative beta cell sources that we could use? Well, porcine donors have been considered as a source of islet cells for a number of years, also for other organs for transplantation, such as kidneys. What has been done internationally? Well, people have isolated insulin-producing cells from fetal pigs. It's an extremely simple manufacturing procedure. It's scalable. However, these cells are functionally immature because they're from fetal pigs. Similarly, with neonatal pigs, it's simple, it's scalable, and they're also, however, functionally mature. Adult porcine islets, it's a much more complex manufacturing process. They're more functionally mature because they're adult. However, internationally, not many people can isolate high quality islets from adult porcines. So there's been a lot of work working with neonatal pigs. So what has actually been done in patients transplanting porcine islets? Well, years ago in 1994, Carl Groth from Uppsala, Sweden, transplanted fetal insulin-producing cells into about 4 patients with type 1 diabetes. They just did not put a therapeutic dose in. They transplanted these fetal pig islets underneath the patient's kidney and managed just to do safety studies and show that some of the cells survive. So porcine insulin-producing cells have been in patients. There's also a study that was done in New Zealand with Living Cell Technologies. Robert Elliott did this. They did quite a few transplants in patients there, and it was only a safety and efficacy study. And from what I understand, they showed that it wasn't that efficient, but it had no detrimental effects on the patients at all. There's also a company in Japan, Diatrans. They have transplanted neonatal porcine islets into patients with type 1 diabetes. And they have, however, a lot of these studies done outside of North America in these countries, they're undergoing very limited amount of safety data. So it's really too bad because they are doing these clinical trials with porcine islets, but they're lacking to show the community what their safety data is. So my lab developed a method to isolate a large number of insulin-producing cells from neonatal pigs. We have corrected diabetes in mice. We've also corrected diabetes in adult pigs and non-human primates. So we feel like we have a good product that we could potentially go into patients with type 1 diabetes to treat them. So what is the clinical translation? Well, when we did the Edmonton protocol, we know in human clinical islet allografts, when we transplanted and we're able to get patients off insulin, each patient received approximately 3 million insulin-producing beta cells per kilogram body weight. To manufacture this many beta cells to treat a patient would cost about $40,000 in manufacturing cost. Talking to people from ViaCyte, Novo Nordisk, to manufacture this many insulin-producing cells to treat one patient would cost significantly more money, about $400,000 to treat one patient. So that's really limited on what you can do because it's an extremely expensive process. So our feeling is that neonatal pig insulin-producing cells may be a good therapeutic approach for treating patients with type 1 diabetes. So what are the limitations as facing porcine beta cell transplantation? There's the current safety concern with porcine endogenous retrovirus. Are we going to transmit that to these patients? Do the donor pigs have to be pathogen free? And there's also the requisite for long-term systemic immunosuppression because we'd probably need even more immunosuppression to prevent rejection of a pig insulin-producing cell than from a human cell. So these are currently our obstacles. And the main obstacle also is if we could overcome these, what would be the regulatory requirements to treat patients? So what is the issue with PERV? Well, George Church has a company in the Cambridge area where they're using the CRISPR technology to genetically modify pigs that do not have PERV. So they're also putting a lot of genes in there that would prevent rejection. So with the advancement in CRISPR technology to generate genetically modified pigs, we may be able to generate insulin-producing cells from these CRISPR pigs that one won't express PERV and two might not be rejected or use less immunosuppression to prevent their rejection. So we are making advancements in the field and hopefully one day porcine cells would be able to be transplanted to treat patients with type 1 diabetes. So I'd like to thank my collaborations with Andrew Pepper and my funding from different agencies. Thank you very much for your attention.
Thanks, Greg. That's fascinating and I think a really exciting and fast-moving area of type 1 diabetes research. And thank you also for making up a little bit of time so that we can stay almost on schedule. So if you stop sharing, I will now introduce our fourth speaker and final speaker in this session. Is Dr. Bassam Haidar, who is manager of the Metabolic and Musculoskeletal Drugs Division at Health Canada. He will be speaking on advances in the treatment of severe hypoglycemia. And I'd like to highlight for anybody listening in French through simultaneous interpretation that a full French version of this slide deck is available for download from the online program for anybody that would prefer to follow along with the slides in French. And I will post the link to the web agenda so that you can access those direct from online. So welcome, Bassam, and I'll invite you to share your presentation now. We need you to unmute, Bassam. We still can't hear you. So I think you're unmuted, but we still have no audio. No, nothing yet. All right. Maybe your headset is not working. Still no.
It may be that he has to select a microphone.
Do you hear me well now?
We hear you well, yeah.
All right. Okay. I think this is a problem with the headsets.
This is good. We hear you fine.
Okay, let me share my presentation. Just let me know when you see it, Sarah.
Not yet.
I just share it right now.
I still don't see it. If you click share screen and select the PowerPoint, it should pop up.
This is what I'm doing right now. Are you able to see it? Just let me know.
No, not yet.
Sarah, it's Pat. Do you want me to share it from my screen?
I also have it here if you'd like us to share it for you, Bassam.
Yes, please.
Okay. Pat, I will let you do that actually.
Because I see it actually in my screen.
Strange. So Pat Tohill is supporting us on the technical side, and I'll invite him to share those slides.
Okay, thank you.
So if you just say next slide, yes, we see that now. Thank you.
Do you see it now?
Yep. Go ahead.
Okay. Great. Thank you very much. I'm sorry for this glitch. I would like to present today my topic on the advances in the treatment of severe hypoglycemia. Next slide, please. Okay, I have nothing to disclose. So hypoglycemia is defined as a condition of decreased blood glucose level that expose a patient to potential harm. Obviously, the most common acute complication it is in type 1 diabetes and also occur in type 2 diabetes. In this context of pediatric population, type 1 diabetes is the predominant risk factor for hypoglycemia. I've been told last week that we have parents who live with a patient with type 1 diabetes, and when parents discover that their kids diagnosed with type 1 diabetes, and putting on insulin daily, they discover also they have a second problem, which is hypoglycemia. So hypoglycemia, it's potentially life-threatening, and for child and a scary experience for family. Some of the causes of hypoglycemia are listed on the right side, particularly in children, in red, wrong dosing, skipping meals, or strenuous physical activities. Next slide, please. So this study highlights that the incidence of hypoglycemia in adult patient population is quite high. In a global 4-week prospective study in adult patients treated with insulins, 83% of patients with type 1 diabetes and 46% of patients of type 2 diabetes reported hypoglycemia. In the next slide. In the, just please go back one slide. In the Canadian cohort of this study, the incidence of hypoglycemia is quite higher. Is about 95% in patients who have type 1 diabetes and 64% patients with type 2 diabetes. The incidence rate is about 128 patient events per year. That is approximately every 3 days, a patient with type 1 diabetes experience event of hypoglycemia. Next slide, please. So this is hypoglycemia can be categorized into a mild, moderate, and severe, and it is associated with the plasma glucose drops. The severity depends on the symptoms. In mild hypoglycemia, where the glucose is less than 4 millimolars, you can have autonomic symptoms such as, dizziness or swelling, nausea. But in the severe case, you can have a severe severe coma, and also this can be cause death. And typically, the glucose level is less than 2.8 millimolars. Next. This is again a retrospective study highlight the risk of hypoglycemia. As you can see, 40% of the respondents experience at least one episode of hypoglycemia, and in this study they highlighted the risk factors, which is it could be patients taking insulins or patients have A1C less than 7%. Next slide, please. So we saw that hypoglycemia can be severe. So how we can prevent that? I think there has to be a combined effort from a patient, from a caregiver, and also from us, Health Canada, to tackle the severe hypoglycemia. Diabetes education is the most efficient and economic way to resolve hypoglycemia. And also, we heard this morning for the medical device, we have Health Canada approved a couple of glucose sensing, for example, Dexcom G6, Freestyle Libre, and these are good medical device to measure a glucose level. And also, the third piece, it's therapeutic products. We have a lot of drugs with less potential of hypoglycemia. We have the insulin analogs and also metformin, DPP4, SGLT2s, and GLP-1 receptor agonists. Next slide, please. So let's focus on the next slide. And we saw, no, that's okay. There's a slide slow there, but that's okay. Let's focus on the education. Yeah, you can go one slide more. I think you have to go back. Sorry, I have to go back. Okay, thank you. So here we saw hypoglycemia can be severe, and so education is important for physicians and families to have to be educated for risk factor for hypoglycemia, but also reporting side effects to Health Canada. It's helpful in doing that. Health Canada can make these drugs safer assessing these adverse event. Also can send safety alerts. For example, so to make to make these drugs safer, for example, in the last 5 years, we have received a report of hypoglycemia. As you can see here, almost 700 cases of hypoglycemia have been received so far. 95% they are severe cases. 75% fatality rate. As you can see, 73% of they are adults, but still 20% of these report the age was not reported. So reporting good side effects and reporting is very important. Among these, you can see also almost 2/3 were insulin dependent. Next slide, please. Next slide. So we have next slide. Yeah, we have seen that patients can have good reporting is important, but also it can change the prescribing practices in somehow for certain the drug in the diabetes area. And I want to show this slide because this is important to show the trend, actually, to see what's going on in the prescribing information of these diabetes drugs. And we can see that in blue, the insulin's purchases in Canada over the five years. You can see in cyan blue right there, there's an increase of long-acting insulins. And also what you can see that the metformin at the bottom in olive color for DPP-4 class remain constant, whereas there is a decrease of the sulfonylurea in purple color, and also an increase, maybe in the next slide you can see it more, sorry, go ahead. Okay, go ahead one slide more, please. Okay. So what you can see, there's a yearly decrease of sulfonylurea over the last 5 years, and this is a class of drug. We know that there's a potential of high severe hypoglycemia, and this is countered by a yearly increase of the GLP-1 agonist receptor or the SGLT2's inhibitors. So there's a sweet success for these two classes because GLP-1 receptors have been known that they have less nocturnal hypoglycemia, and also they have a potential for weight loss management. SGLT2s who've been very busy the last 5 years to work on these indications of this class of drug for other indication regardless of diabetes. And I've been saying this is drug class. It's not only for diabetes, but for cardiology. Sometimes 40% of reduction of heart failure. This is amazing. So not only for heart failure indication, but also the cardiorenal protections indication they have right now. So you can see the fantastic trend. Next slide, please. Just go back one, please. So we have seen a class of drug. They have in a patient they can have an option to have less drug with less potential of hypoglycemia, but I think glucagon still remain the main treatment option for hypoglycemia. It's supposed to be happening. Next slide. So glucagon is the first-line treatment for severe hypoglycemia. In fasting state, it is secreted by pancreas to boost glucose level in the liver. And next slide. Next slide, please. And when insulin is secreted, actually, it blocks the secretion of glucagon, and reduce the simulation of reduce the secretion of glucose in the liver. In type 1 diabetes or advanced to type 2 diabetes, we have a defective in this mechanism. Next slide. So glucagon is really a good drug for treatment of severe hypoglycemia, but it has to be, the administration is a little bit complex, as you can see in the picture. It has to be mixed, need a needle, mixing beforehand, and that's why I don't think this is very much used. So next slide, please. So a Canadian researcher, and the father of 2 kids of type 1 diabetes, discover a new formulation and a compact device to boost nasal glucagon as a simple puff in the nose. Next slide. So the company came to us 2 years ago to approve this drug device combination. It's a powder. It's very simple, very easy to use. Next slide. And we approve it. Next slide, please. We approve it in type 1 diabetes, mainly type 1 diabetes and type 2 diabetes, for the treatment of severe hypoglycemic reactions, which may occur in management of insulin-treated adult and pediatric patient population when impaired consciousness precludes oral carbohydrates. So for the physician we have in this room, the drug device is not for patient taking sulfonylurea, for example, because it gives a false sense of security because of the long half-life of these drugs and the repeated episode of hypoglycemia. Next slide, please. Okay, we don't have to read the whole slide, although this is a very small compact device, but the company came to us with a large comprehensive clinical program, including pivotal studies, confirmatory studies, real-world user studies, and simulated studies, and also the package with toxicology and immunogenicity studies because this is a synthetic protein. And next slide, I'll be focusing my 2 next slides on the pivotal studies and the usability studies. In short, I mean, these are the core of the pivotal studies in patients with type 1, but also in type 2 diabetes. This is a crossover trial where where insulin was given to induce hypoglycemia to a certain range. I believe that less than 2.5 millimolars, to drop the glucose level to less than 2.8 millimolars. And then as you can see at the bottom of the slide, and then glucagon was given. When glucagon was given after the administration of glucagon 3 milligram or glucagon intramuscularly, this is a comparator. Sorry. There is a rapid raise of glucose level within 30 minutes. So it's working. The blood glucose increase rapidly, and it shows that both glucagon, nasal glucagon, is non-inferior to intramuscular glucagon in this setting. Next slide. They did exactly the same, not exactly the same thing. They did, a similar study in pediatric patients population with a different age, different cohort, as you can see there. But they haven't induced hypoglycemia for ethical reason, but they use insulins to drop glucose level before 4 millimolar, I believe. Next slide. As you can see, at the bottom, there is 100% success rate in both formulations, and the glucose level was increased by more than 1.4 millimolar. Next slide. In terms of safety, there's both formulations are comparable in pediatric and adult patient population, and there's a few headaches and nasal symptoms with the nasal formulation. Both formulations, they have some gastrointestinal symptoms, for example, nausea and vomiting. But it just resolved in a few days. Next slide. Not only nasal glucagon works to increase blood glucose level, but also can rescue patients with severe symptoms of hypoglycemia. This is a real-world evidence studies. The patients and caregiver taught to how to use the nasal glucagon device and register the time to awake from severe hypoglycemia events, and also monitor glucose level. In this study in children and adults, and the hypoglycemic events were resolved within 30 minutes, and administration was very easy, less than 30 seconds. There was 12 severe cognitive impairment with loss of consciousness, and nasal glucagon was able to rescue this patient population. Next slide, please. To summarize, nasal glucagon was comparable to intramuscular glucagon to rescue patients with severe hypoglycemia. In terms of safety, both formulations are comparable, and this is first time we have seen a combination drug device that is very easy to use, convenient for the patient, for the caregiver, especially for kids. Next slide. I would like to add my presentation with this slide. The Flame of Hope sits in Banting Square in London, Ontario, and the flame will be only extinguished when a cure of diabetes is found. Thank you very much. Next slide. Sorry about this glitch.
No problem. Thank you, Bassam, also for that very poignant closing slide. I would love to visit the Flame of Hope one day and look forward to the day that we can extinguish it. So thank you all to the speakers for your wonderful presentations today. If our speakers could turn their cameras on, and I'm inviting everybody to either raise a hand in the Zoom function, which you can find by looking at the participant list. And if you can't find that, you can give a thumbs up or some other icon to indicate that you'd like to ask a question verbally. And finally, you're also welcome to ask questions in the chat. So while we are waiting for a couple of questions to come in, there's a question I'd like to put to all of the panelists. And for Bassam, this is a little different for you because you talked about something that has already been approved. But if you could name one thing as concisely as possible, what is the major barrier that you see to advancing the work that you talked about to people living with diabetes today? And that could be funding, it could be regulatory issues, it could be more safety data, anything goes. So I'll call upon you in turn and please be concise so that we can get to other questions. So Bruce, I'll start with you.
Yeah, I think I spoke around adjunct drugs, and I kind of highlighted a class that has some little accentuation in diabetic ketoacidosis. So to me, the biggest gap is just the existence of DKA and the fact that in present clinical care, we've not handled that well enough. So we have education, and for example, we know pumps increase DKA risk, so we have extra education around DKA management. 6 months later, I'll see a patient who would start on a pump and had a sick day, but couldn't remember the concepts around ketones or wouldn't have measured. So somehow our whole system needs to do better with the kind of really basic fundamental aspects of type 1 diabetes management to begin with. And that's going to open the way to more and more, being able to incorporate more and more therapies, including adjunctive drugs.
Okay. Thank you. Thank you, Bruce. And look forward to lots more data coming out around adjunct therapies. We're excited to know that there's several studies ongoing in Canada now. So Ahmad, how about you? What do you see as a key barrier to moving closed loop therapies and adjunct therapies forward?
Yeah, maybe a couple of things. I don't think we can do much about them, but it's the reality. So one of them, because in my field I need to collaborate with companies, and that is drug companies, algorithm companies, sensor companies, the drug companies. And for some of these companies, maybe they either don't believe in your approach, or they're small, they don't have enough resources to support you. So my dependency on sometimes up to 5 companies for a single project sometimes can delay things for me. Like even buying pramlintide, for example, I had to spend 6 months to find a way to get it from the US and so on. So sometimes it's just because the companies have no interest, basically. Now this is one thing, but I don't think we can do much about it, because companies, their priority is try to bring their products to market. Like it's not to try to help researchers, and they have their own research. And maybe the other thing is that things take a lot of time, and I don't think there is a single issue that you can point my finger at and we solve it. Like contracts takes time, RAB takes time, regulatory reviewer takes time, buying material takes time, finance, HR. I mean, every single hurdle takes a month, and collectively it ends up adding 12 months overhead, and I don't think we can do anything about it. At least, I mean, I learned to live with it. So these are not complaints, but these are just generally what sometimes I wish they were different, but I think they're fine.
Thank you. I think those are really important points. And for sure, JDRF's aim with our research is to accelerate progress. And it's an extremely complex challenge because of the many different stages at which we have to work to make that happen. So I completely hear you. And I'd love to talk more about that specific to your work and other work in that area. So Greg, on to you. If you could name 1 or maybe 2 key barriers you see to moving the field of cell replacement therapy towards the clinic.
Well, I think beta cell replacement therapy would be a really good physiological way to obtain good glucose homeostasis. But the way the field is right now is we've got to devise strategies to prevent rejection. And because the amount of immunosuppression these patients have to take, there's too many risks associated with the benefit. So I think if we can come up with ways, and there are, and JDRF is funding some of those strategies. So I think that's the first thing. And then the regulatory issues related with putting porcine cells into patients. I think there's been movement in the field for many years using pigs as sources of cells and organs. But it's the regulatory part, and that's a bit of a hurdle. But first, we have to make sure we could do this safely. So, okay.
Thanks, Greg. And on to Bassam. So please take this question as you wish. Perhaps you'd like to comment on what were the barriers that were overcome to bring nasal glucagon through to approval. So open to your perspectives.
I think for glucagon was in good collaboration between us and the company beforehand. And I was listening to Dr. Perkins on the moving beyond certain drugs in type 1 diabetes. I think we can do a lot to refine the patient population, because obviously there's this class of drug given to type 2 diabetes, they're not investigated much in type 1 diabetes. So I heard that talk, and I heard we approached discussion with this company about how to use certain class for type 1 diabetes, but it has to be well done. It has to be for example, Dr. Perkins was talking about the DKA risk, and then it's something that we care about. Not only have this class of drug on the market because of efficacy, but sometimes the risk, it's overwhelming, and we need to fine tune the population. And I appreciate that the European approve these drugs in certain subgroup over overweight, over 30 in type 1 diabetes. But us and FDA were pretty much, we asked the company to provide more data. Sometimes we find that the companies, they want to promote their drug right away, but we encourage them to work with us to further assess these new classes of drug in type 1 diabetes. And second, I would like to see, I haven't presented data on the clinical trial application. I have seen clinical trial application over the 5 years decline in Canada in type 1 diabetes. I would appreciate if we can have more patient populations, so especially Canadians, and because Canadians, they have a higher rate of diabetes, type 1 and type 2. I wish I had more data on the Canadians data set and more engagement from researchers and sponsor companies to run clinical trial in type 1 diabetes. And a third, I wish that also we can do a lot of things together. I wish we can have early discussion with the company at the early stage of the development program, that we can remove some obstacles and to gain efficiency and to remove some of the regulatory barriers. If we can have a conversation with them early on in the clinical development program, as Dr. Corbett said.
Thank you. We are nearing the end of the session, so I'll do a final call for questions. I don't see any hands or icons. We have a quiet crowd today. Bassam, if I can ask you one more quick question with a very short answer, if you wouldn't mind. We talked a little bit about different regulatory decisions in different countries, and I wondered if Health Canada is working towards greater coordination and working with other regulators so that we can find efficiencies and possibly move to approving new devices and drugs more quickly in concert with other countries. I know that's a big question, but if you could speak to that very briefly, it would be helpful.
I appreciate the question. I think before COVID-19 situation, we already engaged with our partners internationally, and I would agree that these connections and collaboration are very fruitful. We can share information between colleagues, FDA and the Europeans, but I can give you an example. We have right now what's called the ACCESS Consortium program, comprised of Canada, Australia, Singapore, and Switzerland, and for new active substance. And the companies will submit new drug submissions simultaneously to all these 5 countries, and we can share the labor in drug review. So we'll for that we can help to reduce redundancies. We can gain efficiency and promote alignments with other jurisdictions.
Thank you. That's really helpful. I regret we need to close the session to be mindful of everybody's time, so I apologize for the abbreviated discussion. We welcome all feedback and further questions if you'd like to submit them after the event. And finally, we have a closing video to share with you from Celia Lorenko from Health Canada and Dave Proughton from JDRF. It's just a couple of minutes long. I'd be very grateful if you could stay to hear their closing comments, after which the session will close. So thank you very much for everyone attending and bearing with us with our technical challenges, and especially to our speakers today for wonderful presentations.
Thank you.
Thank you very much.
Thank you.
We don't have any audio on the video. We're trying again.
Good afternoon. It's my pleasure to be here to do the closing remarks to this remarkable session. I would like to first say a big thank you to all the distinguished speakers for sharing their knowledge and experience with us today. We spoke about a number of really important elements from the management, treatment, and management of type 1 diabetes all the way to the prevention of type 1 diabetes. JDRF's motto is
Okay, yet another technical glitch, but I imagine we will reboot that in a moment. Okay. I think if we have the audio, that's...
to co-host this session today. Good afternoon. It's my pleasure to be here to do the closing remarks to this remarkable session. I would like to first say a big thank you to all the distinguished speakers for sharing their knowledge and experience with us today. We spoke about a number of really important elements from the management, treatment, and management of type 1 diabetes all the way to the prevention of type 1 diabetes. JDRF's motto is from type 1 to type none. And that's why we got here together today in order to discuss how can we get to that vision of type none. I would like to also take this opportunity to say a big thank you to JDRF for allowing us to co-host this session today in order to delve into these important elements of the treatment and management of type 1 diabetes. These patients face all sorts of challenges on a daily basis, regardless of whether there is a pandemic or any other issue. And that's why
Okay, we've lost the video again. I think we need to close the session.
Canada's
So
very much interested in continuing these conversations as research advances and treatments emerge, so that indeed we can turn type 1 into type none in the future. Thank you, and wishing you all a wonderful rest of your day. Thank you.
As Bringing Breakthroughs to Life is coming to a close, I want to thank everyone that was involved in making today's event such a tremendous success. I want to thank Health Canada, our co-host, all of our attendees, the moderators and presenters, for giving of your time because it made today such a rich experience. You helped us deliver a symposium that created dialogue, knowledge exchange, and you certainly saw firsthand lots of new and emerging treatments for type 1 diabetes. And I'm convinced that with ongoing collaboration and partnership, we can ensure that people with type 1 diabetes will live healthier, easier, and safer lives. And ultimately, we will be able to give them freedom from this relentless disease. On behalf of everyone at JDRF and the type 1 diabetes community in Canada, thank you for joining us today.